Hi, my name is John Irwin. I'm a contributing writer for the Yale Scientific magazine. And today I'll be talking with Joel Rozowsky, Yale Scientific Researcher. Joel, could you take us through your statistical analysis of John Rind's microarray data?
Sure. Yeah, so we worked on this project that got published a while back in the developmental cell, studying the molecular differences between males and females. So the experimental setup for the analysis, in order to identify the molecular differences between boys and girls or men and women, was we used the technology of the time, which was gene microarrays. And a gene microarray basically has on a microarray oligonucleotide that measure the expression of about 20,000 genes in the human genome or the mouse genome simultaneously. And the experimental setup is we got RNA samples from male and female for a variety of different tissues from multiple males and multiple females in order to be able to statistically significantly assess the differences between male and females for all the genes or most of the genes in the human and mouse genome. And so the experimental setup is basically on a gene-by-gene basis. You can take the measurements of expression for all these genes between all the samples between all the different individuals, males and females, and you basically look for genes that show that and most of the genes end up having the same expression, so you don't see expression difference between the males and females, and a small subset you can assess the statistical significance of whether you see an expression difference between males and females, and that's basically the experimental design.
All right, cool. And can you go a little more into detail about your statistical work with it?
Sure. Yeah, so one of the expertise that, my expertise is the sort of more called bioinformatics, is basically to do a sort of robust statistical computational analysis on the sort of high throughput data that comes from these sort of biological high throughput experiments. So when the experimentalist does these experiments, they do the hybridizations, they run the machines, they scan the arrays, and they basically send to me a set of files with the data output of the arrays. And the format is for each sample, you get a file which corresponds to each of the genes on the array and a corresponding measurement of the expression, the degree of which that gene is expressed, how much RNA is present in the cell corresponding to that gene. Okay, so from my perspective, in order to do the statistical analysis, the first step is basically make sure that the arrays are normalized to each other. So there's a normalization procedure to make sure that the sort of average intensity and sort of peculiarities of how much material was hybridized to the array are sort of normalized out, so that you can do a fair statistical comparison between the different samples, between the males and the females. And after we do the normalization procedure, then we sort of compare, as I said, on a gene-by-gene basis, genes that show differential expression between males and females, and one could do that for each of the different tissues. So basically identify in a statistically rigorous and a conservative fashion which genes show differences between males and females for both, as I said, human and mouse, between males and females for different tissues. And one can do this, and we use, as I said, relatively conservative statistical parameters, and you get the output is basically a small list of genes that show differential expression, and you can separate in those that show expression that's high in the female or high in the male, because they could go in either direction.
All right, thank you very much. How significant do you believe the molecular differences are between boys and girls?
Well, I think for the small subset of genes that we found to be significant, they are statistically significant, meaning you could do the experiment again and you'd find, they'd be robust. The experiment would be reproducible. However, the count, the number of genes that actually show differential expression between males and females is a really small fraction of the total number of genes that are present in the mouse and the human. I mean, on average, mouse and human have about 20,000 genes. And for the tissues that we investigated, we found of the order of 20 or 30 genes that showed robust statistical significance. So I think, in general, there's not that many things that show differences, but those things, depending on the role of those specific genes in terms of the proteins that they make, they could have a very important roles. And because obviously men and female, for the most part, function very similarly, but in certain the key areas are significantly different.
Thank you. Since you graduated grad school in 1998, how have you seen the field of bioinformatics change?
So one thing that has changed dramatically is that when we did this analysis, the state-of-the-art technology was using gene-based microarrays. And this was called high-throughput genomics at the time. And basically, you did a microarray study, and you were able to measure the gene expression for 20,000 genes in one experiment at one time. And at that time, that was sort of an amazing experiment. As you know, over the last sort of almost 10 years, what's sort of gone from gene-based arrays where you had to know the genes that you were interested to whole genome arrays where you basically could tile a whole genome using oligonucleotides. So you didn't need to know where the genes were and what was being expressed. And then, so it went from like 20,000 measurements in a single experiment to 1 million measurements to a single experiment to now we use sequencing-based experiments, high throughput. So in a current high throughput sequencing experiment, experiment that can that can be run in like 2 or 3 days generates the equivalent of like maybe 100 million reads. So the amount of data that one generates from these sort of experiments has dramatically increased. So the sort of the necessity of the computational side of the analysis, the bioinformatics, has become a lot more intensive. And, you know, this is something that sort of traditional wet lab type biologists have a hard time and need assistance in terms of doing the analysis.
Very good. Where do you see the field of bioinformatics heading?
So, as I said, one of the big developments in the last couple of years is the sort of advent of sort of high throughput genomics, and the scale of the sequencing and the type of data one can do has dramatically sort of opened up what one can analyze. So before, you know, when one started doing human genomics, we had the reference genome. So they sequenced the reference genome for one representative individual. So now one thing that's sort of becoming very thing is rather than look at one individual, is sort of doing personalized genomics. So looking at the sequence of you or me and sequencing like everybody and sort of identifying how functional genomics like RNA expression or, you know, gene expression, all this sort of stuff relates to a single individual and how you compare to the rest of the population. So sort of this population genomics stuff is the sort of the current future direction, I think.
Thank you. And also, would you be able to talk a little about your current projects?
Oh, so one of my current projects is we're involved. I'm in the lab of Mark Gerstein, who's a professor in MBB, and we're part of this big ENCODE consortium. So the ENCODE consortium stands for Encyclopedia of DNA Elements. And basically the role of ENCODE is to identify all the functional elements in the human genome. So protein coding genes or genes that make proteins are pretty well annotated in the genome. But there's a whole sort of menagerie of other types of functional elements in terms of what does the sequence of the human genome do, in terms of binding sites where transcription factors bind in order to regulate genes, in terms of other things that are expressed that don't code for genes that are called non-coding RNAs. And basically the sort of project is in order to fully classify the entire sequence of the human genome in terms of what it does. And I had a conference call earlier today. We're in the processes of sort of publishing a whole, issue of Nature and other journals in order to sort of publish the results of this project. And this should be coming out in the next couple of months.
Awesome. Thank you very much.
Cool. Thank you.